Today topic of discussion LE cells, LE bodies and LE phenomenon. First coming to LE bodies. What is LE bodies? Anti-nuclear antibody cannot penetrate the intact cell. What is intact cell? The intact cell is complete cell which has the nucleus and everything. Intact cell have everything, nucleus and everything in it. Here the cell we are talking about the damaged DNA. So the anti-nuclear antibody cannot penetrate the intact damage to DNA. But when the nuclei are exposed, this anti-nuclear antibody fuse with the damaged DNA antigen. When the antibody and antigen fuse, it form antigen antibody complex. Anti-nuclear antibody cannot penetrate the intact cell because this one is the damaged DNA. But when nuclei are exposed, anti-nuclear antibody fuse with the damaged DNA antigen form antigen antibody complex. Here condition, when nuclei are exposed, that time only it form antigen antibody complex. This complex formed, this one name is called LE bodies. It is also called hematoxylin bodies. The antigen antibody complex which is formed is the LE bodies. So what is LE bodies? It is a fusion of anti-nuclear antibody and a damaged cell nuclear antigen. So fusion of this anti-nuclear antibody and damaged cell nuclear antigen. The fusion of these both creates an LE bodies. Now it is called as hematoxylin hematoxylin bodies. Why? Because it is basophilic and it is observed the hematoxylin stain. Why we are calling hematoxylin bodies? Because it observed hematoxylin stain. What is LE cells? LE cells are leukocytes. Mainly this neutrophil and macrophages which engulf these LE bodies or LE cells. Leukocytes, neutrophil and macrophages which engulf these LE bodies. This one name is called as LE cell. So the neutrophil and macrophages that engulfing the LE body is the LE cell. LE cells are microscopic finding, microscopically found in systemic lupus erythematosus, but not specific to systemic lupus erythematosus. This can also be found in pericardial, pleural fluid and also in case of rheumatoid arthritis, etc. So this one found in systemic lupus erythematosus, pericardial, pleural fluid and rheumatoid arthritis, etc. Now coming to the picture. First one, this one is the normal neutrophil. It has tri-lobed nucleus. Tri-lobed nucleus placed centrally, where it is placed in center, and these are the granules, inside granules also. Now coming to LE cells. What is an LE cells? It is a cell which engulf the LE bodies. Middle portion which you can see able this violet color one. This one is the LE bodies engulfed by neutrophil. Now this neutrophil with engulfed LE bodies is called LE cell. What are the changes you can see these LE cells? First thing, tri-lobed nucleus fused. Here it is presented in center. Now it is fused to that periphery because the central part is taken up by that LE bodies because central part Le bodies is present. Now coming to Le cell phenomenon. So Le cell phenomenon, the principle behind this phenomenon is anti-nuclear antibody fused with damaged DNA nuclear antigen. Uh, Anti-nuclear antibody fused with damaged DNA nuclear antigen. That means antigen antibody fusion only occur when the nuclei are exposed. Coming to procedure, first we have to exposure that nuclear antigen. So when it is exposed, this fusion occur. So for the exposure we need to do it by the rotatory bead method. When we use this rotatory bead method by continuous rotation by the glass speed, this nucleus get exposed. So when exposed it form antigen antibody complex, that means Le bodies. Now Le bodies are enveloped by Le cells. These Le cells are seen under microscope. Commonly there are the microscopic finding of SLE. So this is the first diagnostic test for SLE historically. So in this video we discussed Le cells, Le body, then difference between normal cell and Le cells, then Le cell phenomenon. We can observe under microscope first diagnostic test for systemic lupus erythematosus historically. Thanks for watching.